Hello and welcome everyone to this PHI webinar. We're happy that you joined us for this talk on the topic of how label-free single-cell tracking reveals unique cell behavior. Studying heterogeneity in cell culture is a central topic, not only important to tumor studies. Some of you listeners might try to find out how to spot outliers or subpopulations in your cell cultures, or maybe you're trying to discover unique cell behavior across your kinetic cell growth, morphology, or movement data. Traditional methods to monitor individual cells have shown to come with a range of drawbacks on the cost of the cells themselves, on your time and resources, or even on the quality of the data by not providing you with the full picture. However, new technologies are there to revolutionize cell research, and label-free single-cell tracking is one of them. During this webinar today, we will get to see the potential of tracking cells over time in various research areas. We're going to have a look at the drawbacks or the challenges with traditional methods, and you will get an introduction to the label-free and automatic single-cell tracking assay with the HoloMonitor live-cell imaging and analysis system, and how it makes for an easy assay for any cell research lab. My name is Lisa Bodley, and I will be the host for this event. Within our team at Phase Holographic Imaging, short PHI, my work centers around digital and communication, while also being actually a passionate cell biologist. And with me here today, let me introduce to you our speaker and PHI Product Specialist, Lisa Lindström. Hey, Lisa.
Hello, everyone. Nice to meet you.
Let me introduce Lisa just in a couple words. You have been a dedicated cell biologist for like 15 years with your PhD focus on tumor biology. And here at PHI for the past 3.5 years, you have helped scientists to advance their cell research using the HoloMonitor system. Great. Before we get started with your talk, let me highlight some practicalities. First and foremost, please do ask questions. We want to hear from you. You can submit your questions at any time using the chat function in the control panel. And after an about 30 minutes talk, we are going to have a Q&A session at the end of this webinar. And any question we cannot squeeze in there, we're going to answer directly by email. And with that, Lisa, get ready to share your presentation.
Okay, thank you, Lisa, for the introduction, and thank you everyone for joining our webinar today. As Lisa mentioned, today we're going to talk about single cell tracking and how you can use it to monitor your cells. My aim of today's webinar is that we will address the following topics. First, we're going to talk about what is single cell tracking and why you should use it in your research. Secondly, we will learn about the HoloMonitor system and how it works in a swift technology fly-through, and also why you should use it to image and analyze your living cells. Lastly, we will go through some conventional techniques for tracking cells and compare them to how tracking works using the automatic HoloMonitor live cell assay. As a bonus, I will be showing the latest real-life user examples from publications with the HoloMonitor system. As you can see in this movie of MCF7 cells growing for 24 hours, different cells behave in different ways. Some move a lot, some only a little. Some divide, whereas others don't. You can also see how the cell morphology changes when the cell moves or divides. To be able to study these cellular behaviors in more details, researchers use single cell tracking. So that brings us to the first topic of this webinar. What is single cell tracking? Single cell tracking is to follow a cell over time and studying what is happening to them during that time, both when it comes to cell behavior, like how they move and divide or don't divide, but also how they change in morphology. As you can see in this movie of MDA-MB231 breast cancer cells, some cells move more than others, and some cells are also thinner or thicker than other cells. Depending on the method you're using, this can be done both on a cell population level, and here are some examples. The top graph showing mean cell speed, the middle graph showing accumulated cell distance, the cells have moved, and the bottom is the mean cell volume over time, or changes in cell volume over time. But of course, tracking can also be done on single cell level. And here is two examples. The top graph shows a cell movement plot where each line in the plot represents one individual cell and how that cell has moved during the time lapse. Another example is in the bottom plot, and that shows how cell volume has changed for one cell family. The sharp dips in the curve represent cell divisions. The examples shown here are all outputs from the HoloMonitor system, which gives you data on both population and single cell level. Then we come to the next question, and that is, why is it important to know what the cells are doing, and why should you use single cell tracking to find out? Normally, when researchers study cell fate, like proliferation, cell cycle, or cell death, it's common to use endpoint assays, such as MTT, WST, or caspase assay, meaning that you compare the status of your cells at a few time points during the experiment at the best, and sometimes only at the beginning and the end of the experiments. This experimental setup comes with a range of drawbacks. First of all, the researcher loses information on what's happened to the cells between the measurements. That is, they lose the temporal resolution. Secondly, these methods require quite a lot of manual work, and this could introduce the risk of human errors, which could lead to less reproducible results. Thirdly, many of these assays are indirect methods. That is, they don't measure what you actually want to study. For example, MTT measures the mitochondrial activity, but is used to correlate cell proliferation. And as you only get average data on the cell population by using this method, you also lose the information of subpopulations within your cell population, and cell heterogeneity. Lastly, in these methods, the cells are used. That is, they are killed or lysed during the analysis, and they can't be used for further downstream analysis. In comparison, if you would use single cell tracking, the researcher would get the whole picture of what is happening to the cells during the experiment. It will not only give you cell number and cell divisions at the end of your experiment, but it can also tell you how long it takes for a drug to have effect, or how the morphology change in response to a certain stimuli. As you could see in this movie of HeLa cells, some cells divide more frequently than others, and this can be used by the user to study cell heterogeneity and subpopulation in more detail. Another example where researchers could benefit from using single cell tracking is when studying cell movement. Again, many of the traditional methods the researchers use to study cell movements are endpoint assays. For example, transwell and wound healing assays, also called scratch assay. These endpoint assays come with a similar set of drawbacks to those you use when studying cell fate. Like, you lose the temporal resolution and the work process is manual, which introduces the risk of human errors and reduced reproducibility. But in the case with studying cell movement with endpoint assay, I would say the biggest drawback is probably that you don't measure what you want to study. Because in an endpoint assay, the researcher will only get the information on the migration of the cell. That is the distance a cell has traveled between A and B. But often the motility, that is the actual path the cell has taken to travel the distance between A and B, is what is more interesting to study. So if you, instead of using endpoint methods, use single cell tracking, you can also study cell motility, which will give you the directness of movement and also more accurate measurements of cell speed. A perfect example of this is the commonly used wound healing or scratch assay. Normally, researchers use only a few images with long intervals over the time of the experiments, and then they can tell when the gap is closed. And by these images, they cannot tell exact time the gap closed or how the gap was closed. Was it closed by the cell movement, cell proliferation? Did all the cell move as a collected front, or were there some forerunners? And as you can see in this movie from the HoloMonitor wound healing assay, all these questions can be answered using single cell tracking and the HoloMonitor system. Here you can see that the cells move pretty much as a collected front, but there are some forerunners, and there are also some cells that divide. There are, of course, also kinetic methods that are used to study cell behavior, cell fate, and movement, but many of these include introducing fluorescent stains or labels to the cells to be able to track them over time. As you can see in these example images, here are UTOA cells expressing Cas9 CRISPR GFP, and they are imaged for 4 hours to be able to track their morphological changes. Fluorescence in turn introduces another set of drawbacks. First of all, there are two major drawbacks with using fluorescence, which is phototoxicity and photobleaching, which are limiting the time the samples can be imaged and the frequency of imaging. Secondly, when adding a dye or a stain to the cell, either by transfection, uptake, antibody binding, or for example, diffusion, the cells become modified in a way that can affect their behavior. Also, some cells are very difficult to transfect and label, such as stem cells and primary cells. There are, of course, ways to image cells without fluorescence, like phase contrast and DIC. And these techniques come with their own set of drawbacks, where the most pronounced is the difficulty in quantifying the data collected. We touched upon this topic of studying cell heterogeneity in subpopulations earlier, and here I just want to show you an example from a publication where the authors used tracking to get information about subpopulations, which then can be specifically useful when studying, for example, drug effect and resistance. In this publication, Zhang et al. Evaluated the HoloMonitor motility assay, and with the help of single cell tracking found a subpopulation of cells that behave differently compared to the average cell populations. In the cell movement graphs on the left, you can see a very small group of cells that move faster and longer distance than all the other cells. They would go undetected using methods that give only average results. With these slides, I hope I have managed to show you the value of single cell tracking and what that can bring to a wide range of research areas. And now we're going to switch topic and get to know the HoloMonitor system a bit better. So during this part of the webinar, I will talk about digital holographic microscopy, which is the technology used in the HoloMonitor system. For those of you who haven't seen the HoloMonitor in action before, it is a small cell culture microscope designed to operate 24/7 inside your standard incubator. Without the need for any labels or stains. It uses digital holography to record and quantify your cell culture in real time. HoloMonitor is paired with its own software, offering a wide range of live cell assays, all having the same straightforward assay setup and following you through automated image analysis, resulting in result presentation and data export. Here we see a screenshot from the kinetic morphology assay as an example of how the software looks. I will now just shortly explain the technology behind the HoloMonitor system. And as I mentioned, it's based on digital holographic imaging. This means that we have a low power laser beam going into the system through the back, where the laser beam is divided into a sample beam that travels through the cell sample and the objective, and then one reference beam that travels unhindered through the back of the instrument. After the objective, the two beams are merged again before hitting the image sensor. What happens to the light as it passes through the cells is that the cells will slow down the light and create a phase shift compared to the reference beam. The phase shift is then recorded by the image sensor, and with the help of sophisticated computer algorithms, the computer calculates and reconstructs the image into what we see when looking at the images in the software. As the light passes through the cells, no light energy is absorbed by the cells, which makes the system non-invasive and also allows HoloMonitor to acquire time-lapse sequences over extended periods without compromising cell behavior. And this properties makes it perfect for single-cell tracking. If you want to know more about the technology, you can watch our on-demand webinar, Label Your Tubes, Not Your Cells, or Rediscover Cell Shapes. The technology does not only allow you to visualize the cells, but also give you quantitative data on all cells in the image. As you can see in the top left image, there is a line covering the five of the cells, and underneath is a histogram showing the thickness across the line. As you can see, there is a sharp contrast between the cells and the background, which makes it easy for the computer to segment the cells. This is achieved because we have the information of how much the light is delayed in each pixel of the image, which is then translated by the computer into height measurement, and using this, the software can identify what is background and what is a cell. If you compare this to how a similar measurement would look in a phase contrast image, you can see that there is no clear profile separating the cells from the background, which, as I mentioned earlier, makes these images very hard to quantify. Once we have found the cells, we can get a lot of data out from each individual cells. We can, for example, look at a multitude of morphological features. Here are an example like area, thickness, volume, irregularity, and textures. We can also look at the cell profile. But we are not limited at looking at only morphology. If we have a sequence of images, we can also study cell fate and cell movement. And the best thing of all is that this is achieved right inside your cell incubator without the need to add any stains or labels to your cells. In fact, all the colors in the image that you see are added in the software to represent the thickness of the cells. One of the nicest things with HoloMonitor is that all information is stored in the captured image of the cells. Therefore, you can reuse your image in the results from one experiment to get multiple results using any of our other live cell assays. This is a powerful tool as it will save you time and save your cells, as you don't have to set up several different time and cell-consuming assays for each assay result you wish to get. That is, we put you and your cells first. To summarize why you should use HoloMonitor to image your cells. The cells are imaged in a non-invasive way. No labels or toxic stains are needed. You can run it automatic live cell assays 24/7 directly inside your standard incubator. You will get real-time quantitative data on both single cell and cell populations. And also, you can generate multiple results from one experimental setup using the reanalysis functionality and save time with an intuitive software and guided workflow that is developed for cell biologists by cell biologists. Now that we covered why you should consider using single cell tracking and the technology behind HoloMonitor, we will continue with how single cell tracking actually works. I will start by going through the general workflow and then compare it to how single cell tracking works in the HoloMonitor. Tracking cells with conventional imaging techniques, such as phase contrast, DIC, or fluorescent imaging, can be a time-consuming process. And depending on what you're interested in, might require some preparation. For example, expressing a fluorescent dye or marker. And this is especially true if you're interested in studying morphological changes. And as we talked about some slides back, this comes with a set of drawbacks to consider when setting up the experiment, such as phototoxicity, photobleaching, and the risk of affecting cell behavior. Once you collected the images, you often have to transfer them to a third-party software and start the analysis with some image processing, for example, background adjustments. And then you can continue with tracking the cells you're interested in. And depending on the quality of the image, this process can require more or less manual work. Once you have tracked the cells you're interested in, you need to take the raw data and create and analyze your results. On the other hand, HoloMonitor Single Cell Tracking Assay need no special preparation of the cells before imaging. You don't even have to move the cells outside the incubator. You just place the cell sample on the HoloMonitor and follow the guided setup for the assay and start to image your cells. Once the time-lapse is completed, the software identifies all cells in the image and creates a segmentation mask, which separates the cells from the background. Then the software automatically tracks the cells in the image and create a multitude of results for you to explore. And of course, it's also possible to export all the raw data if you would like to explore the results even further. So compare general single cell tracking and HoloMonitor Single Cell Tracking Assay. With the HoloMonitor Single Cell Tracking Assay, you don't require any preparation of the cells, and it's completely non-invasive. The imaging is set up in a user-friendly software that guides the user through the setup. Once the imaging is complete, the cells are segmented and all cells are tracked automatically, which means there is no need to transfer your images to third-party software. Also, HoloMonitor software automatically generates a multitude of results for you to explore as soon as the tracking is done. But you still have all the raw data available for further analysis if you would like to. So to summarize this part of the webinar, why should you use HoloMonitor Single Cell Tracking Assay to track your cells? You increase your efficiency with automatic single cell tracking, and it tracks all cells in an image. HoloMonitor study individual cells non-invasively, so there is no need to add stains or labels, which save the cells for further downstream analysis. You can explore both individual cell and cell population behavior over time within the same experiment, as well as monitoring both changes in cell movement and morphology in the same experiment at the same time. Lastly, it will save you time with an easy-to-use software. Now we will continue with looking a bit closer into the different outputs HoloMonitor Single Cell Tracking Assay can give you. For example, cell movement and cell morphology. And this is an example video from the HoloMonitor system where you can see how cells move, but also morphological changes. The first output feature I would like to show you is the movement plot. In the movement plot, you can see how the cells have moved over the course of the time lapse. And in addition to just showing how the cells have moved, this will also give you data on, for example, cell migration, motility, cell speed, and the directness of each individual cell. In this example, you can see that many of the cells move in a similar way, but there are some cells that move quite differently from all the others, giving you information about cell heterogeneity and subpopulations. I'm now going to show you a couple of examples on how our users have used the movement plots in their work. The first example I would like to share with you is from a publication by Chirumarry and colleagues that used the HoloMonitor to investigate how antimicrobial peptides affect the motility and migration of breast cancer cells. And as you can see in these graphs, the addition of peptide lowered the motility and migration of the cells compared to DMSO-treated cells. In the next example, I would like to show you Szatanek and colleagues have performed a chemotaxis assay using the HoloMonitor system to determine the effect of tumor-derived microvesicles on cell motility. The next type of output you can get from the HoloMonitor Single Cell Tracking Assay is cell feature plot. This gives you the chance to study changes in morphology of the tracked cells over the course of the time lapse. Some of the most used morphological features include volume, area, irregularity, thickness, and textures. And in this movie, we are following the cell outlined in blue at the top of the image and looking at how the volume changed over 24 hours. The sharp drop in volume occurs every time the cell divides. HoloMonitor software is, however, not limited to only these 5 output features, but contain more than 30 different morphological features to choose from, depending on what you're interested in studying. And an excellent example how these feature plots can be used is the publication by Croft and colleagues. Here they used the HoloMonitor to quantify morphological changes after treatment with different drugs. And by looking at how the roughness changed over time, they could distinguish between cells undergoing mitosis and dying cells. Lastly, I would like to show you the latest addition to the output parameters in our software, namely the cell tree plot. Thanks to a new algorithm, the latest update of the single cell tracking assay, we can now create these cell trees automatically. In the cell tree, all cells are grouped into cell families, and this gives you information such as cell cycle length, number of divisions within a cell family, and information of cell fate. Here in the movie, each cell family is represented by its own colors, and the fork in the plot represents the division. In a recent publications, Wallin et al. Used HoloMonitor to study effect of externally added Cystatin C on A375 melanoma cells. They looked at proliferation, cell survival, number of mitosis, and doubling time, and they could see that externally added Cystatin C reduced growth and increased cell cycle time in these cells. Another example from our users is this publication by Yumol and colleagues where they used HoloMonitor Single Cell Tracking assay to study how combinations of different drugs affected number of cell divisions and time spent in mitosis. If you're interested to look at even more HoloMonitor publication, you find over 175 peer-reviewed papers on our website. I would also like to take a minute to mention that we recently released a major software update where we have improved the single cell tracking assay based on feedback from our users. I would like to remind all our current users who are listening today to download it and try it out. And if you're not yet a user but are interested in finding out more, to contact us or to book a personal demo through our website. So, to sum this webinar up, why should you use HoloMonitors to study single cell tracking? Because with the HoloMonitor system, you study both individual cells and whole cell population behavior over time, which can help you to study cell heterogeneity or subpopulations. Because with the HoloMonitor, you image cells in a non-invasive way while not compromising on quantitative data, but at the same time circumventing many of the drawbacks with conventional methods. Lastly, because with the HoloMonitor system, you increase your productivity by learning more from every experiment, by extracting multiple results, which will save you time and save your cells. Lastly, I would like to end this webinar by showing you this video highlighting an example of how HoloMonitor single cell tracking can be used to study effect of drug treatments on cell movement. Here you can see the control cells that move considerably more compared to the colsamide treated cells.
Thank you, Lisa, for your talk. I hope that all of you listening enjoyed this overview of single cell tracking, the HoloMonitor, and see how it can be a powerful tool to study individual cell behavior. I, by the way, want to remind you that we actually have attached 2 handouts to this webinar where you can find more information about the HoloMonitor system and the single cell tracking assay protocol. Both you can actually also download from our website. And now it is time to answer your questions. Lisa, are you ready?
Yes, I am.
Okay, if you have one or multiple, if you haven't submitted them yet, please ask them using the chat function. Or if you have some later on, just contact us via email or go to our website. All right, let me try to rephrase this. Which cell types did you show again in the videos? Are there, maybe it's like a question towards like many like other cell types?
Yeah, so in the videos, I mainly showed different breast cancer cell lines like MDA MB231 and MCF7. But I also showed some HeLa cells, but I guess you can summarize it as it is mainly cancer cell lines. But of course, many more cell types can be imaged with the HoloMonitor system. And HoloMonitor is especially good when working with sensitive cells like stem cells or primary cells. It's a non-invasive imaging and analysis tool and you don't require any stains or labels to be able to image your cells. In our publication list on the website, you can filter the publications by cell type, so that could be a way for you to find out more about the cell types used with the system. And if you're curious to know if the specific cells you are working with have been used together with the HoloMonitor system, you can always contact us and we will find materials and answers for you. But in general, you could probably say that the HoloMonitor system is great for monitoring adherent cells and monolayer cultures, but within a broad range of applications.
That was a great answer. I hope that this answer was answered. This question was answered. And as you said, if there's anything more, just reach out to us. All right, there is another one. See if I can rephrase this. Can I cell spend? So if you run the HoloMonitor Single Cell Tracking assay, are the cells spent, or what can you do with them afterwards? Can you use them again for additional downstream assays? Can you say something about that, Lisa?
Yeah, I understand. It's a great question. Yeah, well, since the HoloMonitor Single Cell Tracking assay is completely non-invasive and you don't need any stains or labels, the sample can be reused in downstream experiments using any type of instrumentation. As an example, I could give you can start by using the single cell tracking assay to study cell movement or to identify a subpopulation of cells that react differently to, for example, a drug treatment. Then you can reuse that data, for example, in the HoloMonitor cell morphology analysis to dig even deeper into your results. But then later, after your time lapse is completed, you can reuse the sample, the same plate, to continue with either stainings or, for example, looking at gene expression or any other type of assay you would like to do.
So yeah, they're staying untouched, and so they're not used in that sense, so you could reuse them.
Yes, absolutely.
It's one of the great features. Let's talk, there were some questions about sort of the export, export of data. So we'll pick those two up. Can I export the raw data from the HoloMonitor system?
Yes, of course. All the raw data, along with the pre-made graphs, movements, plots, and cell trees can be exported to Microsoft Excel, where you can analyze and present the data in whatever way suits you. And there you can also, if you want to combine, for example, 3 repeats of your experiments, they can be combined here to generate more extensive statistics, for example.
So you look for the data in the software, but you can then use it also afterwards for further analysis and statistics. I think that might have been the question, so it's good. The other question was not about the data, but actually about the images and movies. So how does that with the export?
Of course, you can export the images and movies. The export of images and movies from the HoloMonitor software, you can choose from many different file formats, including JPEG, TIFF, PNG, MP4. And as you might have seen, some of the images are black and white and some are colorful. And you can choose the color sets for your cells in whatever way you want. So you can generate your own color sets, and this will be exported together with the images or movies. And you can also view the cells either in 2D or 3D. You can zoom in to a certain part of the image or use the whole field of view.
Yeah, especially those 3D cells. You see a lot on our social media because they are pretty.
Yes.
But yeah, no cells were harmed in the process, right?
No, exactly. It's all colors added afterwards in software by the computer.
Let's pick one more. And if
Yeah.
your question was not answered yet, we will get back to you like as soon as possible. Let's pick. Yeah, this one came in quite early on. About the, I think that means field of view. How large is the field of view? It's a quite common question we do get, so maybe just let's answer that.
Yeah, so the field of view is 500 by times 500 micrometers. But you're not limited to one field of view per well, so you can put up multiple positions within one well to get a larger representation of what's happening in that well.
Maybe just to elaborate on that, like that was just something I just thought of. When you speak of well, like they're probably vessels.
Yes.
How about that? Different vessels compatible with the system.
Yeah, so the HoloMonitor supports use of a range of different vessels, from chamber slides to 35 millimeter Petri dishes, 6-well plates, 24-well plates, and 96-well plates. And as always, if you have any particular questions about a vessel, you can just shoot us a question by email and we will get back to you regarding that. But I would say that the most important thing here is also like that we can put in, we not only image like one position per well, but we can actually do multiple positions per well to get the larger sample imaged.
Yeah. Okay, let's wrap it here for today. Thank you for all your questions. I know there's still some in the list. We will get back to you by email, so don't worry about that. You're always actually welcome to always reach out to us. You find here on this slide our website, viap.com, or you can also shoot us an email. The contact details are on the website as well. To stay up to date with what we are up to and the latest in live cell imaging, I just want to give you a shout out. Please follow us on social media, engage with us. We always love doing this. And with that, thank you everyone joining us today. Thank you, Lisa, for your talk.
Thank you.
Until next time. Yeah. Bye.
Bye.